Hi everyone, welcome back to the channel. I'm the GCSE science teacher, and in today's video we're going to be learning about cloning in plants. I've recently done a video where I talked about cloning of animals and the ethical considerations and the process of this, so I'll link that for you either in the cards or in the description box. I hope you find this video helpful. Let me know if you do, and let's get started. So first of all, why would anybody want to clone a plant? I can understand some people think it's quite sci-fi and cool to be able to clone an animal or a human in theory, and wouldn't it be amazing to have someone to go and do various tasks for you if you had a clone of yourself and things like that. It's all cool to think about, but why would we want to clone plants? Well, the reason for this is because actually, if we want to look at food security, we want to look at certain crops for medicines. Remember, plants are very much the origins of medicine. And also with climate change, we want to increase the amount of plants that are on the earth because of deforestation. There are lots of reasons why plant cloning would be beneficial. And often plant cloning follows on from other technologies like genetic modification, which I have covered on my channel and I'll link for you in terms of genetic engineering, that is. And this is because if we can artificially create plants that are disease resistant or pest resistant, we can actually clone those plants, and this can increase the number of those plants that we can then sell or we can then use for farming techniques and agriculture. So really lots of uses that we can think about. But how is it done? Well, the first type of cloning we could do in plants is a very simple one that really anyone can do, and you may have actually done yourself before. And this is just by taking a simple cutting of a plant. So often we see this in things like strawberries and also in spider plants as well. If we just take a small section of a plant, we can actually clone it. So how is this actually done? Well, a branch from a parent plant is cut and the lower leaves are removed. And then the plant is dipped into damp compost and some hormones can be added to encourage new growth. However, this in reality, you don't always need to do this. You can literally just take a small cutting and plant it in a pot. But for the sake of GCSE, they like to add in a few extra steps because this is what would be done on a commercial scale. So the hormones are used to encourage that new growth. The plant would then be kept in a moist environment and new roots would begin to develop, and then we'd be able to create that new clone and it would have the same genetic properties as the parent plant. This is a much cheaper, much quicker method than the one I'm going to show you, which is called tissue culture. And this is one that's often done in smaller scale farms or smaller scale gardens as well. And like I say, this is something that you yourself could do if you were interested in this. But in terms of much of a larger scale way of cloning a plant, we have this idea of tissue culture. Sometimes this is called micropropagation, and this just means exactly what it says. So micro meaning small, and propagation just means to increase the number of. So micro, we're talking about the small plants that form and then obviously create larger ones. So the first step in tissue culture is as follows. So scrapings of the tissue itself from the plant are taken from that desired plant, and explants are transferred into sterile agar gels, which contain a growth medium, which is a mixture of different hormones and nutrients. So before we move on, an explant is just the small section, small tissue taken from the plant, the scrapings of that plant. And some of those hormones that we've discussed before on this channel include auxin. This encourages growth of the cells that are there. Remember, those cells are most likely going to be meristems, those stem cells that are found at the roots and shoots of plants. Now, those explants are grown in vitro until they form very small cells called plantlets. And those plantlets are then transferred into nurseries until fully grown. Now, the word nursery we're talking about is just a small patch of land, a small area of soil which can be nurtured. I've put in bold all the keywords you need to know, and the words plantlets, explant, in vitro, these are all words that they like you to use whenever you're describing tissue culture or micropropagation. In vitro, remember, is just a Latin word for in glass. That's talking specifically about the sterile agar, and it's sterilized because we don't want any competing microorganisms that could affect the growth of those explants, but also contaminate the culture and introduce diseases and things like that, which we do not want. So these are the different ways that scientists can use essentially cloning in plants to create lots of different copies of the same plant. And this can preserve a lot of rare species specifically, and species of plants that we don't have many samples of anymore. So this is actually quite a useful technique. So with any of these technologies, we also have to think about the ethical considerations. I've mentioned. Although the welfare of animals is higher than, say, for plants, people are still concerned about the effects that plants, if they are cloned, could have on our ecosystem, on plants around them, the species that are living around those plants, the animals that consume them, including ourselves, and the implications that could have on the wider climate and, like I say, the wider ecosystem. We also need to think about the effects on the environment. Could these plants take over other neighboring species? Could it lead to human cloning again? Because as technologies develop, other ideas come to the forefront, and then this could drive different ideas. We also need to think about morality and reducing genetic diversity. All of these are really key points to consider. And how they could ask you in GCSE to discuss this is perhaps they would give you a for or against argument. You would then have to use that information to come up with your own idea and evaluate the technology and give your own conclusion. You might be asked to discuss how cloning in plants is done and compare it to animal cloning. You could be asked to just write the stages or to explain what plant cloning is or what a clone is. Lots of different questions they could ask. One of the other things to consider as well, just in terms of this slide as well, I've just said here that tissue cloning is much more expensive than just the plant cuttings that I mentioned before. However, the positives of this, like I said, it does preserve those rare species that we obviously want to encourage and maintain. So, especially with the changing climate that we know of at the moment, this is actually quite an important technology if we want to preserve those species on Earth, especially in areas that are quite remote, like the Galapagos Islands that Darwin visited back in all those hundreds of years ago. There are lots of things on Earth that are slowly diminishing because of climate change. So actually these technologies could prevent that from occurring. And that's it from me today. I've been the GCSE science teacher and you have been curious. If you did enjoy this video, feel free to give it a thumbs up. Do share it with someone else so they can learn more. And of course, subscribe if you haven't already. Join this amazing community. And if it sparked an interest in yourself about certain questions you may have about this topic, feel free to comment them down in the comments section. I'll also link some interesting articles and YouTube videos about cloning and plants and technologies that I've found and you might also find interesting as further reading. In the meantime, have a great day and I will catch you in the next one. Take care.